So this video is on DNA profiling, and what that really means is how we can use DNA information to make a unique profile or fingerprint for an individual, often used in forensic science or paternity testing to look at relatedness between individuals. It's different from sequencing, so sequencing looks at every single nucleotide in order to sequence a whole length of DNA, which also can be used for those things. DNA profiling just looks at specific markers within that stretch of DNA. It's quicker and easier, but still provides a high degree of reliability. One of the key terms within this is electropherograms and electrophoresis. So electrophoresis is a process used to separate pieces of DNA based on size, and an electropherogram is a graph that comes from that process that allows you to look at bands of DNA, where they are and how intense they are. Basically one of the things that's really a starter on this is that when you look at the amount of DNA within a human cell, it's enormous. So this is a different way of looking at chromosomes. Here's some chromosomes within the nucleus, and so these are in the unwound state. So several different chromosomes there, but these are huge molecules of DNA, as you can imagine. And so it's not really easy or practical to analyze the whole sequence of DNA. Instead, we just want to look at parts of that, fragments of DNA, just little snippets really, and they give us enough information in order to do our analysis. So one extra thing we're going to add to our molecular toolkit now and start thinking about is what we call restriction enzymes. Restriction enzymes are special enzymes, proteins that catalyze a reaction, and the reaction that these ones catalyze is cutting DNA, breaking the covalent bonds in DNA in order to cut DNA up into smaller pieces. It will only cut at a specific sequence. So if we have a look at this diagram here, this is one of the first restriction enzymes that was discovered and characterized. It's called EcoR1, and EcoR1 looks for the sequence GAATTC, or obviously on the corresponding strand is CTTAAG, which is actually the same as the top one backwards. It's a special sort of case there. And what it'll do is it'll cut between the GNA and this strand and the GNA and that strand, leaving two pieces that are separated. Obviously this overhanging region there that's in the maroon, it's possible to sort of connect those up by base pairing, and they could rejoin, and that's what we call the sticky ends. So these two T's and A's combined with those two A's and T's could join up again through hydrogen bonds, and so this is how the DNA could come back together, and it would need a ligase enzyme to join those up. But the key thing to remember here is restriction enzymes will cut that DNA. There are other restriction enzymes other than EcoR1, so HindD3 will cut at this sequence, leaving 5' sticky ends. PST1 cuts this sequence, leaving 3' overhang sticky ends. And EcoRV does a blunt end cut, leaving like this. So the amazing thing about this is that these are all different bacteria that the restriction enzymes have been isolated from. So EcoR1 comes from E. coli. Each bacterium has its own special suite or collection of restriction enzymes for cutting up the DNA of viruses that attack them. And so biochemists have been able to isolate and identify these, and now we have sort of a library of different ones. So whatever sequence we're looking for, try and find a matching restriction enzyme that can cut at that sequence, and therefore we can cut DNA in specific places. Not everywhere, of course, but wherever those sequences are, match it up with the right restriction enzyme, and you can cut DNA up into pieces. So that's a key one there. Once you've cut DNA up into pieces, though, you've got these fragments. So you end up with a test tube with a mix of whole different DNA pieces. That doesn't really help you. That's just the first step. So the next step is what's called electrophoresis, and that allows you to separate those pieces based on size. Also, just as a reminder, either you've cut up the pieces or you could use PCR to make lots of pieces of DNA. Either way, you're going to be putting those fragments through electrophoresis to separate them out by size. So extraction, cutting up and loading onto electrophoresis. You can then separate those out, and I'll talk about that on this other section here in a minute. and then you're able to visualize those separated pieces in what's called a DNA fingerprint. So here's our mixture of cut up DNA, different pieces. DNA has a negative charge, and so if you put it in a gel with an anode and a cathode, it will be repelled by the cathode, attracted by the anode, and run down that. Now if there was no gel, it would just quickly go from one side to the other. What the gel does is it slows down those pieces as they go through. Small pieces can move faster through the gel because they're less restricted. So the small pieces come down faster and end up at the bottom, and the large pieces are up here. Just one note, I want this to be really clear to you. These bands, what we see here as blue lines, are not pieces of DNA. What they are is it's a whole lot of pieces of DNA together. So that band would be thousands of pieces of DNA that are all in that location, and when you identify them through this sort of X-ray or fluorescence, you will see that as a band, but that's not suggesting that's a piece of DNA, that line. That line is representing a band or a clump of DNA that's all moved together at the same time. So because each person has those unique differences in their DNA, when we cut with the restriction enzyme, it might cut at different places, or because we've got parts of DNA that are longer or shorter, even if it cuts at the same kind of places, the pieces might be different sizes. And so you end up with a pattern of different pieces of different sizes, and that's what we refer to as the profile or the fingerprint. So any sequence differences would lead to the change of the size of fragments and a change in the pattern of bands. So one way we can identify this and analyze this is using the electropherogram. So here we might have alleles. So remember, when we're talking about genes, different versions of an allele, different versions of a gene are called an allele. This allele 1 has a section in one of the introns that has 5 repeated segments, and allele 2 has 11 repeated segments. It's just a bit of junk DNA that's in there in the intron, but what it does is it makes that gene longer or shorter. So when we run that through the gel, the short piece is going to come through first, the long piece is going to come through later, and give us 2 peaks. So as the DNA goes through this laser and detector, you get a blip and then another blip. So what we're seeing here in this peak is that's just a representation of a band. As the band comes through the detector, you get a blip and then another one. So these 2 different sizes then are an indicator of 2 different alleles. And so we can start to see how electropherograms are a way of looking at the genotype of an individual, the different alleles that are at that locus or that location in the DNA. And you put a few of those together. At this locus, you've got one with 13 repeats and one with 16. At this locus, 28 and a 29. At this locus, 2 copies of 9, so that one's going to be the homozygous one. And at this locus, an 11 and a 12 repeat. So this is what the repeats look like. Something like a GATA gets repeated 8 times, 9 times, 10 times. And this just makes that segment of DNA longer or shorter, leading to 1 or 2 blips coming through on the electropherogram. These are then unique for individuals. If you just looked at a few locations, there might be a few people who have the same, but if you look at lots of locations, 16, 20 locations or whatever, this pattern becomes a unique identifier of that person, their DNA profile. So this is covered in Chapter 5, and there's a couple of videos there that you might want to watch as well.